of scientists warns that screens are broadly harmful, while the others argues that such concerns are exaggerated, but yet the lack of consensus remains regarding all fields, definitions, conceptualization, and classification. Key questions also includes causality of problematic internet use. Also, considerable heterogeneity exists across measurement scales, markers, and proposed mechanisms, as majority of search of biomarkers of problematic behaviors are really highly inconsistent. But on the other hand, what we know from large studies, they consistently show links between digital use and well-being, with positive associations with depression, anxiety, loneliness, and all the related difficulties. And when internet use looks problematic, it also usually tends to appear problematic on mental health measures. What else we know that evidence also points toward cognitive processes, and many executive functions such as inhibition, conflict detection, working memory, among many others, they also appear to be involved, with various alterations reported in problematic samples. And when we do not have clear biomarkers, conceptual debates also remain unresolved. And what I mean is whether problematic internet use is best understood as an addictive or impulse control or obsessive compulsive disorders. And multiple frameworks have been applied, but currently leaning to addiction concept, because symptom expression, such as, for example, heightened reward sensitivity or craving states, they do overlap with substance use disorders and other behavioral addictions, and which is also reinforced by neuroimaging findings. Yet, most of this knowledge from things what we know for sure, all of this is drawn from heavy usage cases. And empirical research often treats internet use as some kind of binary state. We have healthy versus problematic internet users. And of course, these distinctions, they do matter because they mark a clinical endpoint, but they tell us little about the majority of internet users, like most people listening here to the conversation, right? Which is regular users, variable in focus, in mood and control, but highly engaged and functioning, not patients. So the question is, how does internet use affect the cognition, mental health, and brain dynamics of approximately 60% of the global population that is online and does not reach clinical thresholds? And that is what we actually barely know. So researchers therefore stress the importance of distinguishing normal habitual internet engagement from truly pathological use, and they do encourage conceptualization of mental health disorders along of normal to pathology spectrum, rather than through this rigid categorical diagnosis. And when applied to problematic internet use, this spectrum approach suggests a continuum of online engagement ranging from healthy to excessive or problematic. And when presenting work of our group, what we did, we actually integrated such approach, not to diagnose, but to step beyond these binary labels and evaluate this continuous variation in internet use, asking whether specific measures scale within engagement. Now, a bit of introduction how all of this works. One of the leading models, such as iPace model, they do frame problematic usage as a result of interaction between core person characteristics and traits, referring to relatively stable individual characteristics. Also affect or emotional processes, where online activities might serve emotion regulation functions and also cognition, so shaping biases and expectations, and executive functions such as inhibitory control, working memory, cognitive flexibility, and all of them leading to a decision to behave in a specific way. So the same online feed can be a tool or a trap. And according to this model, these factors together, they determine whether high engagement remains adaptive or tips into some kind of problematic internet usage patterns. So of course, there are a variety of tools to assess psychological state or personality traits, which are usually measured by self-report questionnaires. When considering perception of internal and external triggers, the interoception approach offers a useful exploratory lens. So broadly, if for some the term is not very familiar, what interoception means is an inward sense of bodily signals. And it guides decisions by signaling the body's needs to maintain homeostasis. What it also does, it also shapes attention, mood, and self-regulation, and may also buffer or bias how we use technology. Because when interoceptive processing is inadequate, approach avoidance behaviors are disrupted, craving regulation and self-control is weakened, and vulnerability to maladaptive behaviors increases. And that's at least what we know from substance use disorders. And to assess cognitive functioning and associate brain activity patterns within any kind of problematic behaviors, electroencephalography or EEG, I believe, as everyone knows in the conference, it presents a non-invasive and affordable and real-time recording of electrical brain activity under both rest and task-based settings. So one of such tasks is the two-choice reaction time go-no-go task, which is a well-established paradigm to assess inhibitory control, and is really simple because what it requires from participants is only to respond to go stimuli and withhold their responses to no-go trials. Meanwhile, EEG provides a temporally precise view of neural processes underlying task performance. So I'll just briefly introduce the techniques we employed in our work before presenting the results. So considering this go-no-go task under EEG recording, so two late cognitive components are most often assessed. So first, N2, which is associated with conflict monitoring and inhibitory control, and P3, reflecting attention allocation, context updating, and response evaluation. Alongside these late components, the paradigm also measures early stimulus processing. So N1 component is considered to reflect early sensory processing, for example, such as stimulus detection, and is also related to attention allocation, while P2 reflects more perceptual processing needed for stimulus identification. And in general, go-no-go is extensively studied and well-validated paradigm, serving as a trustworthy measure, especially of these late processes happening in the brain, because it helps us to dissociate motor execution and response evaluation within go trials from conflict monitoring and motor inhibition during no-go trials. So that's task-based setting, event-related potentials. Besides these task-based paradigms, resting state EEG serves as an index of spontaneous brain electrical activity in the absence of any kind of sensory or task-driven stimulation. So alpha oscillations are considered as a mechanism reflecting neural inhibition and information gating, and is thought to inversely reflect cortical activity and supporting inhibitory function by facilitating the synchronization of large-scale neural networks. Meanwhile, alpha asymmetry refers to the relative difference in alpha power between left and right hemispheres, and is typically calculated as the log transform difference between right and left alpha power values. So alpha asymmetry in general is one of the most investigated markers of brain lateralization. However, research has predominantly focused on frontal asymmetry, leaving parietal alpha largely underexplored. Yet what is known from the literature parietal asymmetry has been linked to personality traits, emotion processing, vulnerability to psychopathology, and many other variables. And last, as already introduced, microstates. So unlike region or frequency-specific EEG measures, the microstate approach, it segments the continuous EEG signal into brief quasi-stable topographies. And what is known that a small set of reproducible topographies explains over 80% of spatial variance, reflecting these momentary global functional states of the brain. And each of these microstates is thought to reflect distinct physiological and functional aspects of cognition. And source localization studies identified 7, though most research focuses on 4 core microstates from microstate A to D, with newer work including a 5th, microstate E. And 3 temporal parameters are commonly analyzed. So, of course, the occurrence rate, reflecting how often a microstate appears per second, the mean duration, referring how long it persists without interruption, and time coverage, which reflects the proportion of time a microstate is dominant or active. And of course, changes within these parameters can signal altered patterns of brain network functioning. However, only a few studies used this approach within problematic internet use field in general, and actually no prior studies have examined whether microstate dynamics or go-no-go task event-related potentials or parietal alpha asymmetry index or interoceptive variables vary within the continuum of internet use severity in regular users. So to address this gap, what we aimed in our research group, we aimed to investigate internet use patterns and their associations with psychological, personality, interoceptive, and neurophysiological correlates in a non-clinical population. So what we did, we recruited 161 participants, young adults aged 18-35. All reported normal or corrected to normal vision, normal hearing, and of course, good general health. And exclusion criteria included usage of psychotropic or psychoactive substances, excepted nicotine, and those with self-considered or clinically confirmed addictions were also excluded from the study. We also additionally asked female participants to have a regular menstrual cycle, do not use hormonal contraception, and all were tested at the same early follicular phase. So in short, we examined a healthy, non-clinical young adult sample, carefully screened to reduce confounds. And the protocol began with participant preparation for the EEG recording session, which consisted of 3 conditions. So eyes closed resting state, alternating resting state, and go-no-go task recordings. After EEG, of course, there was a set of self-report questionnaires assessing internet use patterns and psychological constructs. And EEG data were then pre-processed using standard procedures. And from this, paradigm-specific features were extracted. So namely, event-related potentials from go-no-go task, alpha power and asymmetry indexes from the alternating condition, and microstate parameters from eyes closed resting state. And when performing our analysis, we used this dual approach. So the full sample for a continuous evaluation, and also between the groups categorized as low and high internet users. So just numbers of all the sets. So considering self-report measures, like full sample was retained for analysis, considering EEG data sets, well, what usually happens, the number of participants varies across paradigms due to data loss, either it's inadequate test performance or technical issues. But the samples are quite similar. And to assess internet usage, we employed 2 tools. So PAU questionnaire, so a brief measure of general problematic internet use, and so-called DPAU. So as a complementary tool that provides a look to specific domains, such as social networking, gaming, messaging, entertainment, etc. So why both of these? What we wanted to capture both broad and also domain-specific aspects of internet usage. So participants also completed questionnaires assessing anxiety, depression, and obsessive-compulsive disorder symptoms. We also measured personality traits and interoceptive awareness. And I will not spend long on psychopathological symptoms because these associations with internet engagement, as I mentioned, these are already well established. But as expected, what we saw that more problematic internet usage, it positively correlated with all the measures assessed, either depression, anxiety, or obsessive compulsive symptoms. And among the PU subdomains, the interesting part was that the total score of the scale and social media use, they correlated positively with all psychological variables, while entertainment and video streaming was linked to anxiety and obsessions, messaging to obsessions, and by contrast, gaming and information search with none of the measures. What kind of suggests that mental health correlates may differ across different activity types. Also, when we compared high and low problematic internet usage groups, of course, significant differences also emerged across all these measures. Now, from what was not known before, we next examined interoceptive awareness to assess if insight into the accuracy of one's bodily perceptions map onto these internet use patterns. And several significant associations were observed. So we saw that overall internet use scores were negatively correlated with not distracting domain, and within internet usage domains, only messaging was negatively correlated with attention regulation, emotional awareness, self-regulation, and body listening. And when exploring interrelationships, we constructed a network model, including interoceptive dimensions, personality traits, and these two internet use measures. And the analysis highlighted neuroticism as a key intermediary, a bridge between PAU and interoceptive awareness. So again, neuroticism was not a new finding, but interoceptive variables was a new subject. So within interoceptive states, neuroticism was negatively associated with trusting, not worrying, and with a weaker connection to self-regulation. And neither of internet use measures was strongly linked to most of the interoceptive dimensions, except for a negative association between PAU questionnaire and not distracting domain. And when evaluating this independent contribution, by linear regression, we also saw that three variables, neuroticism, again, not distracting, and trusting were significant predictors of problematic internet usage. So in other words, individuals tending to avoid or distract themselves from bodily discomfort showed more problematic internet usage. Now, moving to the behavioral and neurophysiological level, the auditory equiprobable go-no-go paradigm was employed with 150 auditory stimuli, like we used two binaural tones, one of them assigned to go stimuli, another to no-go, and participants, as usual in such tasks, had as quickly and as accurately as possible to press a key for go stimuli and withhold their responses for no-go. So considering behavioral results from the measures assessed, no significant associations were found between overall PAU scores and behavioral performance, with one exception. And gaming scores positively correlated with go reaction times, suggesting that more problematic gaming behavior use was linked to slower responses. But in overall, the absence of significant correlations within any of these behavioral outcomes indicates that in the sample of healthy young users, PAU was not associated with basic cognitive performance at the behavioral level. At the ERP's level, the topographies within current sample aligned with prior studies using the same task. So as expected and consistently with the literature, we obtained standard distributions. And again, no significant associations or group differences were found between PAU scores and all assessed potentials, but several specific associations emerged. And what we found that negative correlations were found between N1 component parameters and total DPAU score and also gaming domain scores. That suggests that more problematic specific internet use, and especially gaming-related internet use, is linked to faster or more negative N1 responses, pointing towards more rapid or more automatic early sensory processing. In addition, P3 latency correlated with information search domain. So more frequent information seeking was associated with delayed P3 responses and possibly indicating reduced efficiency in response inhibition at neurophysiological level. So taken together, certain internet use patterns may be related to specific ERPs, but they do not appear consistently across the full spectrum. And when considering the literature with previous studies, they consistently reported ERP alterations within problematic internet behaviors. But these findings across different studies, they were not consistent. And absent results within our sample, in this study, they support the literature from that side that ERP alterations may develop gradually and may be more pronounced only at severely expressed problematic usage levels. Now, next 4-minute alternating resting state session was recorded. So 2 minutes eyes open, followed by 2 minutes eyes closed. And alpha power was assessed from frontal and parietal electrodes, and also 4 electrode pairs was used to analyze alpha asymmetry indexes, again, both at frontal and parietal regions. So grand mean spectra showed expected resting state patterns, pronounced alpha peak during which higher, of course, during eyes closed than eyes open. Now, what we found, weak but significant correlations, and internet use was negatively correlated with alpha power in both regions and across both conditions. And no correlations were found between alpha power and any of psychological variables. Then when compared the groups, the analysis confirmed these associations. So high PAU group, they had significantly lower alpha power than low PAU group in both conditions and across both regions. And when considering alpha asymmetry index, internet use scores positively correlated with parietal asymmetry during eyes open, indicating greater right parietal alpha and thus increased left hemisphere activity with more problematic internet usage. And consistent with these correlations, high internet usage, problematic usage group showed higher parietal asymmetry during eyes open compared to a low internet use group. Again, when summing the results, internet use severity was associated with greater left parietal activity, which is linked from literature to higher behavioral approach, but again, during eyes open condition only. Last paradigm recorded under 5 minutes eyes closed resting state. So the optimal number of microstate topographies was determined to be 5, and again, extracted group level maps displayed prototypical configurations closely aligning with literature. And the occurrence of microstate E and its time coverage positively correlated with problematic internet usage questionnaire score. Additionally, significant associations were observed between microstates B, C, and D parameters and psychological symptoms like anxiety and obsessions, compulsions. And when compared the groups, the same two temporal parameters showed significant differences between the groups. So high problematic internet usage group showed both higher occurrence rate and greater coverage of microstate E compared to low problematic internet usage group. So as already discussed, problematic internet usage, it commonly co-occurs with depression, anxiety, and other related symptoms. And in our sample as well, more problematic internet usage was significantly correlated with all psychological measures we assessed. Yet, the specific association between microstate E hyperactivity and internet use, but none of the psychological symptoms, suggests that microstate E may serve as a distinct neurophysiological marker in this context. So briefly summarize all what was presented. The observed link between lower interoceptive awareness and more problematic internet use, which was bridged by neuroticism, may reflect a potential disconnection from embodied self-regulation. And notably, the not distracting dimension, which points to impaired attentional allocation to internal states, aligns with models, such as my previously showed IPACE model, that hypothesize that internet use may serve as a coping strategy to escape real-life distress. Taken together, this co-occurrence of distress and diminished interoceptive awareness may create a feedback loop in which emotionally reactive individuals with limited bodily insight might turn to digital environments, potentially reinforcing these maladaptive patterns. And what happens over time, such engagement could drive neuroplastic changes in the brain. And considering current findings, so in general, N1 component reflects early stage processing and attention orientation, engaging both bottom-up sensory and also top-down predictive mechanisms. And in individuals with more problematic internet use, N1 modulation at some level might indicate altered attentional gating, possibly reflecting either hyperreactivity to external stimuli or less effortful attention switching, which would indicate a bottom-up attention automatization, which is likely shaped by chronic exposure to stimulus-rich digital environments. Then alpha band results further points toward attentional imbalance. So we have reduced global alpha power in more problematic internet involvement, which suggests increased cortical excitability and lowered perceptual thresholds. And greater left parietal activity indicates approach motivation and externally focused attention. And these effects, strongest under eyes open condition, they support the idea of persistent readiness for external engagement. And lastly, microstate E adds to this profile. So in general, microstate E is associated with salience network and normally is negatively linked to emotional and interoceptive processing. Here it showed an opposite pattern, a greater occurrence and greater coverage in more problematic internet users. And again, that's a pattern that may reflect a shift toward externally focused stimulus-driven states at the expense of somatic awareness. So although all these measures are temporally and spatially distinct, theoretically, all of them converge on a broader neurocognitive shift from internally regulated, interoceptively attuned top-down control toward externally driven, reactive, bottom-up attentional processing. And this shift may underlie core features of problematic internet use patterns: psychological distress, diminished bodily awareness, and also uncontrolled engagement with digital contents, possibly leading to disruption of balance between internal and external attentional modes. So I see I'm running out of time last moment to give credit for a team, our brain states research group, a bunch of students missing in the pictures, but the addictive behaviors was my PhD project interest, but the scope of the group's interests are way broader, with the brain under all the research, Professor Inga Grishkova-Bulanova. So I'm leaving here the code for more work of our group, and thanks again for your attention.
Thank you, Dovile. A really great talk and very wide scope covered. So we don't have questions and we are definitely out of time, but a short question maybe I would like to ask. I apologize. So Gabriel, just a second, and I will hand over the floor to you. But just a short question. You mentioned microstate E, which is usually related to interoception. And also you mentioned some specific EEG differences related to left parietal area. So for me, it rings like it's related to some embodiment mechanisms. So do you think that the extensive internet usage may be related to changes in embodiment cognition?
Yeah, and that's actually like from the starting of the project, our first hypothesis were related to impulsivity. That's why we involved go no go task, and we expected actually changes in these later cognitive potentials. And when we saw that, okay, something is happening in the beginning of the task in these early sensory potentials, and then we started to analyze what's happening within resting state. And when we saw this, okay, so parietal with approach, like it's related to approach motivation. And again, microstate E, as you said, like yes, it's related to interoception, to this inward bodily perception, and we see like there's something happening exactly in these variables. So what I was tried to bring in the generalization in the summary of all of this. So of course, as I said, is distinct parameters, but it kind of gives a sense that probably there's something happening in these embodiment mechanisms, what we can start to observe in the brain activity when these problematic patterns becomes more visible or I know touchable. Thank you. Oh, we cannot hear you.
Oh, sorry. Thank you. And our next speaker is Gabriel Auer, who represents Vienna Medical University, friendly lab. We currently have a joint study, which is supported by Austrian-Ukrainian grant. And Gabriel is specializing on brain electrical activity related to different pathological state, like autistic spectrum disorder. And I'm very interested to hear his talk today because he will talk about EEG markers and specifically about delta-beta coupling, which is delta-beta coupling marker, which is not very common. I've just met this for the first time, and I'm very excited to hear Gabriel's talk. So, Gabriel, we are ready to hear your talk. Thank you.
Thank you very much. Can you hear me?
Yes. Mm-hmm.
Okay, thanks again for the nice introduction. I just gotta share my screen.
Okay, we can see your screen. Mm-hmm.
So can you see now my first slide and only the slide, or can you see the presentation mode?
We see in the presentation mode. Mm-hmm.
Okay, then one moment. How about now?
Sorry, I was wrong. It was just a slide. Currently, we see in the presentation mode.
Okay.
I apologize.
No worries. So you see the first slide and only the first slide, right?
Correct. Correct.
Okay. Okay. So again, thank you very much for the nice introduction. And thank you for the opportunity to present the current state of my research project, which I named Delta-Beta coupling and emotion regulation in adolescents with internalizing disorders. As you've already heard, first a few words about myself. My name is Gabriel Auer, and I'm working at the ABC Brain Lab as a doctoral student. The ABC Brain Lab is led by Dr. Lilian Konica, and in our lab, we conduct translational clinical neuroscience research with a focus on so-called neurotherapeutic methods. Neurotherapy refers to methods that directly modulate brain activity, such as neurostimulation or neurofeedback. So these interventions target mechanisms directly rather than only symptoms. And we evaluate these neurotherapeutic methods by assessing them across subjective, behavioral, and physiological levels, and then cross-validate these levels to get more robust results and deeper understandings of the findings we have. So having said that, a few words to our current research. First, the background. As you might know, unfortunately, anxiety and depression are becoming increasingly prevalent among adolescents. These disorders often co-occur or develop sequentially, which implies that they likely share underlying mechanisms. And one of these common mechanisms that supports the development of both symptoms of anxiety and symptoms of depression has been found to be dysfunctional emotion regulation. And since we're interested in interventions that directly target mechanisms behind clinical manifestations, we want to know which neural system underlies emotion regulation so that we might target it with a neurotherapeutic intervention for treating simultaneously anxiety and depression. And the neural system we found is the ventromedial prefrontal cortex, which you can see here. So why the VMPFC? Well, the VMPFC is an important regulatory hub that integrates subcortical and cortical processes, meaning emotional and cognitive processes. For example, it helps to regulate negative emotions through its bidirectional connections with the amygdala. So to put it plainly, the VMPFC or VMPFC activity supports emotion regulation. Furthermore, aberrant VMPFC activity has been found to be linked to adolescent anxiety and depression. So normalizing VMPFC activity might help treating these disorders. And our method of choice to try and normalize VMPFC activity is transcranial direct current stimulation, abbreviated TDCS. TDCS non-invasively modulates neural excitability through weak direct currents that are applied through the electrodes that you can see here, which is a picture of our TDCS device. And based on prior research, we assume that a nodal TDCS of the VMPFC, which increases VMPFC activity, excitability, may support more flexible and adaptive regulation of emotional responses, potentially reducing anxiety and depressive symptoms. And furthermore, there's a significant gap concerning TDCS studies in adolescents, despite promising evidence in adults, in particular with regard to treating depression. So the following study has been developed. Brain stimulation for emotion regulation in adolescents with psychiatric disorders. So this is our current overarching research project, and the study protocol can be openly accessed online if you're interested and want to know more details that I'm able to talk about in this talk. A few words about our study design. We are trying to recruit 108 adolescents between the ages of 13 and 19 years with clinically relevant difficulties in emotion regulation and symptoms of depression and anxiety, which is assessed via self-and-parent report questionnaires and also clinical interviews. The study is ongoing. So far, we have around 75 to 80 adolescents who have completed the study. Our participants are randomized into either an experimental group or a control group, of which only the experimental group will actually receive a nodal stimulation to control the placebo effect. We then have subjective behavioral and physiological measures before the intervention, which consists of 10 stimulation sessions, and after the intervention, and also subjective and behavioral measures 1 month and 3 months after the last stimulation session as follow-up measurements. So this is the protocol we've been doing for the last few years, and now to my doctoral research project. The thing is, since our overarching project is sham-controlled and I'm blinded, I cannot know who is in which group and can therefore not conduct interim analyses of the pre-post data, which means to assess TDCS efficacy, I'll have to wait until the study is concluded. But what I could access right away is the data assessed before the intervention to follow my own research ideas. So, sorry. This was one too fast. What I'm interested in is neural marker of emotion regulation that might also serve as an intervention marker, since we want to improve emotion regulation capacities. So for assessing and interpreting neural activity, I can either associate fMRI or resting state EEG activity with questionnaire and or behavioral measures. And what I found more appealing is resting state EEG for the following reasons. First of all, it fits our design better, since TDCS can directly modulate resting EEG. It is also cheap, easily accessible, it requires minimum participant effort, and can be consistently replicated across studies and labs. Furthermore, it provides context-independent measures of baseline brain activity, which makes it suitable to assess trait emotion regulation. So diving into the literature of resting state EEG and emotion regulation, I found delta-beta coupling. What is delta-beta coupling? Delta-beta coupling, meaning the correlation or predictive relationship between delta and beta activity during rest, has been proposed as a neural marker of trait-like emotion regulation capacity. The idea is that the functional coherence between slow-wave delta activity reflecting limbic processes and emotion generation, and fast-wave beta band activity reflecting the prefrontal control of these emotions, can be interpreted as emotion regulation processes. And in the current literature, high DBC has been repeatedly linked with anxiety symptoms, mostly social anxiety, and also with high cortisol levels. And this is thought to reflect a rigid overcontrol of persistent emotional reactivity. For example, in social anxiety, it has been found that after cognitive behavioral therapy, symptoms improved alongside reductions in DBC. So according to this, high DBC means worse emotion regulation. However, in typically developing children, high DBC has been found to be associated with greater use of adaptive emotion regulation strategies that flexibly and effectively manage emotions, whereas in depression, DBC did not change following CBT despite symptom improvement. So to reconcile these in part conflicting findings, one approach is to interpret DBC as a neural signature of how emotions and control systems are coupled, and not as a direct measure of how good emotion regulation is. So it becomes clear that DBC functions differently across populations. And while in clinical samples, elevated DBC is usually conceptualized as emotional overcontrol, most studies link DBC only to symptoms and not to how people actually regulate their emotions. In fact, there is only one study I know of that related DBC to emotion regulation, which has been done with children, and there are no studies with adolescents. So to address this gap, I investigate how DBC relates to actual emotion regulation and whether this association is moderated by anxiety and depression severity. Now to the methods. First, about the questionnaires. As a primary outcome, we are using the FEEL-KJ, which is a 90-item test that measures 15 self-reported emotion regulation strategies that are either classified as adaptive or maladaptive. For trait anxiety, we have the STAI, and depression severity is assessed via the administration of the 2. Our resting state recordings are done with the same device that we are using for our stimulation, namely the StarStim 32, and we record 8 minutes of alternating eyes closed and eyes open conditions. And I focus on frontal, central, central and parietal regions, as these are usually the regions of interest in DBC research, and to assess whether neural coupling is localized to frontal control systems, which is usually expected, or distributed more broadly. Now, how is I'm sorry, now it works. Okay, sorry for the delay. Now, how is DBC conceptualized? I follow here the approach of Sarah Miruski and her colleagues, which suggests a linear regression of delta predicting beta activity. So delta activity averaged during the 4 minutes eyes closed and 4 minutes eyes open conditions across frontal, central, and parietal electrodes are used to predict corresponding beta activity across the same regions. So the idea is that subcortical oscillations reflecting limbic activity, meaning delta, trigger prefrontal control mechanisms reflected by beta activity. And then we analyze the absolute residuals of this regression with higher absolute residuals of this regression indicating weaker delta-beta coupling, meaning that beta varies more independently from delta, and that the relationship between them is weak or inconsistent. And lower residual reflect stronger coupling between delta and beta oscillations, where beta behaves in a way that is strongly tied to delta, which is interpreted as more rigid or a more constrained interaction between effect reflected by delta and control reflected by beta. So this is also termed or interpreted as overcontrol or reduced flexibility. And in my research, I expect, based on prior studies, that maladaptive emotion regulation, which supposedly underlies both anxiety and depression, will be positively associated with stronger DBC, meaning with more rigid coupling across the sample, with anxiety strengthening this relationship, while the role of depressive symptom severity is examined exploratorily. Furthermore, I expect findings to be strongest in the frontal cluster because DBC is closely tied to prefrontal control networks, and this is also what prior research has predominantly found. Now, to my initial findings. As expected, anxiety and depression symptoms are highly correlated, and maladaptive emotion regulation predicts both symptom clusters. However, what you can see here is that we have a ceiling effect in the maladaptive emotion regulation scale, meaning that a significant proportion of our sample has the maximum value. So this restricts the variance of MER of maladaptive emotion regulation and limits its ability to show associations with other variables, which of course is a problem for my hypotheses. Yeah, here's also a graphic of the strong negative skewness of maladaptive emotion regulation. And indeed, unfortunately, I did not find any significant associations between DBC and MER, but also not between DBC and symptoms of anxiety or depression, which are more evenly distributed. So DBC might just not be a good marker of maladaptive emotion regulation and internalizing symptoms, which is surprising considering prior research. But what is also surprising is what I did find to be significant, namely central DBC predicts adaptive emotion regulation across eyes closed and eyes open conditions. So adaptive emotion regulation increases with stronger delta-beta coordination in the central region. So I found that central delta-beta coupling across eyes closed and eyes open conditions is selectively associated with adaptive emotion regulation. And this is independent of anxiety and depression or age and gender, and not explained by delta or beta power alone, which shows that the effect is coupling specific. Furthermore, the association is only significant for absolute residuals, not signed residuals. So this means it's the intensity of the coupling, not whether coupling is higher or lower than expected, that matters for emotion regulation in this finding. So central DBC can be described as a selective neural marker of adaptive emotion regulation in my sample, which brings us to the discussion and the next steps I'm planning. So I concluded that in my clinical sample with elevated maladaptive emotion regulation, anxiety, and depression, stronger delta-beta coupling or stronger delta-beta coordination appears adaptive for emotion regulation, indicating that neural stability may be more beneficial than flexibility in the sample. And it might also be used to use this finding to use central DBC as an intervention marker in our coming pre-post TDCS analysis. Of course, some open questions remain. For example, why did I find significant effects only for the central cluster, while previous research has mainly found it in frontal areas? I didn't really have the time yet to dive into that to make an educated guess, so if you have any ideas, I'll be very glad to hear them. And why did I not find any effects for maladaptive emotion regulation? Well, one possible explanation would be the significant negative skewness. But it is also possible that this is a result of the specific conceptualization of DBC, which has already been shown to be associated with adaptive emotion regulation in children, but only in frontal and parietal clusters, not in the central one, which might be attributable to developmental differences. So as a next step, I want to conduct within subject correlation of DBC, meaning correlating every minute of delta with every minute of beta for each subject to get individual correlation scores, which I can then associate with other variables. I also want to dive deeper into specific regulation strategies and check if, for example, subscales of the field coyote like reappraisal or rumination are associated with my DBC constructs. And I haven't really thought much about behavioral measures yet, but I also plan to assess, for example, an emotional Stroop task, which we are doing during our pre-measurements with DBC variables. So this is now from my side. I thank you very much for your attention, and I'll be happy to receive questions or comments. Thank you very much.
Thank you, Gabriel. Very interesting talk. I've never heard previously about delta-beta coupling and how it's about the perspectives of using this index. I see no questions, so maybe I will go with one. So we have just 2 minutes. It's pretty technical. Can you please tell for delta-beta coupling which frequency pass filters did you use? Low pass filters and high pass filters. It may be pretty important for using specifically for estimating the features of delta and beta signal. Thank you.
Yeah, of course. Thank you for the question. I've used a low pass filter of 0.1 and high pass filter of 40 Hz.
So the high pass was 0.1, correct?
Mm-hmm. Exactly.
Thank you. Thank you. So, and we have no other questions. So thank you for your presentation. Great talk.
Thank you for listening.
And it's my pleasure to invite Yuqin Chiu. My colleague from Purdue University. So Yuqin Chiu is a specialist in cognitive flexibility. And I usually find the references to her studies in unexpected papers. Sometimes I wonder how wide her ideas are spread along the literature related to cognitive inhibition and cognitive flexibility and working memory. She will actually present today a talk on cognitive flexibility. So, Yuqin Chiu, we are glad to host you, and we are ready to hear your talk. Thank you.
Okay, cool. Thank you for the introduction. Am I seeing the slides? Okay, everyone seeing the slide? Let me me see.
Yes, yes. Mm-hmm.
I see one of your face. Maybe not. Okay, then I'll just, okay, so go back. Yeah, so I will shift gear from the two previous presentation and talk a little bit more basic science laboratory task. Before that, I'll tell you a little bit of a context where that relates to real life. So given the inflation, I imagine that's happening not just in the US, but also in Europe and elsewhere. Prices are rising, and therefore I've been going to many different grocery stores trying to find the best deal. However, what stressed me out is not the price tag at the end I have to check out, but it's more of this self-checkout process. And I'm sure it's getting very popular in all over the world because they're trying to get rid of human, right? And what really annoys me is I can usually be very quick in terms of shopping, getting the items I want from the aisles. But by the time I get to checkout, things with the barcode, I get them ring the price easily. But then when it comes to grocery, a bunch of tomatoes or apples, then I have to shift my thinking to, okay, here's a screen to find a tomato, and then now it's accounting versus waiting, all these different tasks, if you will, I will call it, that I have to switch to. And then let me tell you another experience where I went to a store where there's human cashier. And what's amazing about her is, as I was putting stuff on the belt, she's ringing stuff, and then she can quickly switch between, the barcode type versus the one that require you to find the item, weight them, or count them. And even by the time I put everything on the conveyor belt, and then she's doing that, at the end, she's able to bag everything quickly. So that get me thinking, what's so different between her and me, right? And you could argue that, well, she's been doing that for many, many times, and she's a professional cashier, right? Practice makes perfect. However, what she shows is not just, stimulus response association, repeating that and then getting really good at that, but a little bit higher level, because she would have to go from, say, barcode scanning to something else, different kind of task set for tomatoes, for a bunch of lettuce, or all these different items. She has to switch the appropriate control operation she applies to these items. So that is the question that got me thinking, how do we study this kind of flexibility, cognitive control in the laboratory? So in this talk, I'm going to tell you a paradigm that we developed to study this, and also a bit more about EEG evidence of the process underlying this. So the overall question is, can higher level cognitive control processes, such as task switching, be encoded within the stimulus control association, almost similar to the stimulus response association that we're familiar with, and be subsequently reinstantiated through bottom-up driven retrieval. So that enable the process to get to the response much faster, and therefore her accuracy, her speedy process compared to my lack of this kind of associations. And to study that, I'll have to tell you the basic task switching boring paradigm in the laboratory we have first. So here, what we have is asking subject to look at some pictures and do categorization task. In this example, subject has to see a picture and judge whether the cat in this picture is a living thing versus a non-living thing, and press a key. The kind of task, the kind of categorization task is indicated by the color of the frame. Okay, so this is one trial, and following this trial, either it's the same task, so repeating the same task, doing another living, non-living judgment based on the picture, or they will have to switch to a different categorization task, such as judging the size of the object being as a reference to like shoebox, so larger or smaller than shoebox. So given this very simple task design, the finding has been shown many times and quite robustly that, on the switch trial, your reaction time is longer and your error rate is higher, which means lower accuracy. And this index of switch versus repeat switch cost is often an indicator of requiring control, a little bit more effortful process as opposed to just pressing a key. Okay. So based on this paradigm, we then make a twist at the item level. We make it such that some of the items are associated with more frequent repeats. So those are the items with low switch probability, item-specific probability as noted here, versus other items that we made it so that it's occurring on switch trial more often. And the color square here is trying to indicate that we also make sure that the item is associated with each task equally often. So it's only the switch probability that differ between these items, not necessarily the task, and because the task will imply response. And so we're trying to make sure the item is associated with something higher level as opposed to be a little bit lower level response. And using this skeletal paradigm, we've been doing different variations. And the finding has been that the item that's associated with high switch probability will incur smaller switch costs compared to items associated with lower switch probability as shown here. And the effect size is not huge. It's 0.22, like a small to sometimes medium almost 0.3. So it's robust in the sense that, a good body of 60 undergrads, we can find this, replicate this finding. But it does take a bit longer trials, longer experiments to find it. Okay, so coming back to this real life example, we hope that using this paradigm, we can then test underlying mechanisms of this cognitive flexibility, as we will call it, using the ISSP effect, as I indicated earlier with the behavioral paradigm. So the interpretation of this effect is that the participant learn item control associations and therefore subsequently can retrieve them quickly and producing this better performance or smaller switch cost. Okay. However, the question that is not answered with this behavioral finding is that how quickly are these item control association being retrieved and how robust they are, right? To some extent, the literature already suggests that this is possible. Dating back from Homel's event file already said that, you can have this stimulus response episode integrated very quickly and then have bears behavioral consequences. And we also know from my own work and other people's work that you can encode or decode task representation from neural signals. But what's missing here, and also combining all that, is a more direct evidence that using EEG, Kyokomoto and Mayer was able to decode stimulus response and rule all together as a conjunctive representation. But what's different we have here is that perhaps we need to also decode the control state association or the switch probability that we use to indicate control state or processes. So the question is, can we leverage the temporal resolution of EEG to decode the high versus low switch probability items, which will tell us whether that representation exists and is useful. I didn't do a whole new experiment just to answer that question. My prior grad student, she already had 2 data sets. collected for asking other questions. But suffice to say that among her design, there are these high-low switch probability manipulations. So we can use her data to address this question. I'll just say briefly how they differ a little bit, but then what's important is that the two data set gave us 3 ISSP conditions, I will call it. So in the first, what I call E1, she had another manipulation that make it 2 sets of the high-low manipulation. But overall, in this experiment, in this study, subject only saw 1 set of high and low switch probability items. Whereas in experiment 2, the subject actually saw 2 sets. So there's 1 set in the E2 baseline condition and another set that's E2 penalty condition. But right now we're just going to treat them as 3 different ISSP conditions for decoding process purposes. But I'll come back to this a little bit later. Before I show you the results, I should show you the typical traditional mass univariate analysis. So here, shown in this top row is 4 different electrodes, all the midline electrodes from the first experiment. And as you can see, if we plot out the time course, the ERP for low and high items, and then look at the difference between the two, there's very little difference. Similarly, in E2 baseline, E2 penalty conditions, you might say there could be a little bit signals here, differentiating high versus low in the E2 baseline, but which is not easily seen here, the error bars are quite overlapping. Okay. However, you could also say, well, I'm only showing you 4 electrodes. What about the rest of us? We do have 64 electrodes. What about the others? So here I'm showing you the E2 baseline across the electrodes topography, activations across different electrodes. And again, high-low eyeballing looks pretty similar. And when you do a subtraction, there's not much difference between the activation across electrodes. Okay, now let's do a more formal statistical analysis. So I'm plotting, using the t-values of the high versus low contrast across electrodes, across time. But when you do this mass univariate analysis, you have to do a proper multiple comparison correction. And this is basically what we have left after this correction called the threshold-free cluster enhancement correction analysis. Same result would emerge if we include a little bit complicated design that we had originally in the experiment, using the full 2x2 in each of the experiment. But suffice to say that from the single electrode, single time point analysis, it's hard to see that we can have decode those 2 items. Okay, so now turning to the multivariate decoding analysis, what we use is a simple linear SVM and just 2 conditions. So the first result I'm showing you is treating each individual trials as a data point, a sample. With that, the training is pretty high, and then test is the one that we actually want to look at. The chance is 50 because there's only 2 conditions, high versus low. And as you can see, a little bit error bar here. So somewhere around here, it's surpassing the 50% chance. If we use a little bit averaging across trials, the classifier does a little bit better. So, but again, slightly earlier, but about a certain time point, past 100 milliseconds, we can start to decode high versus low items. So that's experiment 1. This is experiment 2, 2 different sets, baseline versus penalty, and replicating what we're showing in experiment 1. When we actually figure out what's the time onset of this above chance decoding accuracy, The numbers shown are here. So depending on looking at individual trials or average trial, but suffice to say that decoding is pretty successful. All right, so pause here a little bit. We asked this question, can we leverage temporal resolution of EEG to decode this high versus low switch probability items as an indication of people can acquire stimulus control association and retrieve it to help the performance? And the answer is yes, above chance multivariate decoding accuracy, distinguishing high versus low control stimuli around 130 milliseconds post-stimulus onset thus tells us that there is a rapid retrieval of stimulus control associations underlying this behavior we're demonstrating earlier. Okay, shifting gear to a secondary question, which last time when I gave this talk, I think confused a lot of people. It's a question of how much of this representations is abstract. In my research community, a lot of people want to know if you can acquire this stimulus higher level representation, how can you transfer to a different one, right? So, and in terms of abstractness, if it's truly abstract, then learning one should allow you to transfer to something different. And here we do have this opportunity in the second experiment of my student's prior experiment. She has E2 baseline, E2 penalty, which use two sets of items. So in E2, there are 4 high, 4 low items. And then in E2 penalty, there's another 4 high, 4 low. They are totally unrelated. So for participants, they do have to kind of relearn. And the question here is like how can they transfer, or can they transfer? And that's what we did. We train on one and then test on the other. For example, train on baseline, test on penalty, and train on penalty, test on baseline. And unfortunately, as you can see here, it doesn't generalize, it doesn't transfer very well. So in terms of abstractness, this is not entirely at the level of, you learn once and then you can generalize to all kinds of contexts and conditions. Initially, that question, the finding of this failed cross-generalization actually puzzled me for a while. And then I keep asking my student to show me like their kind of SV and weights back transfer to, they call activation. And after looking at it for a while, I kind of reconcile and I think that, oh, that makes sense. And here's, I'm showing you the best decoding subjects, the map, kind of the SV and weight transfer back the activation map. It's not the mean activation from the electrodes, but another weighted way to look at it. And if you look at the best subject in the two conditions, the topography of this activation looks pretty similar, which would enable this potential transfer. However, when we look across different subjects, it just shows that it's not a consistent kind of representation, and then something that you can learn one and transfer to the other easily. Okay, so to conclude, I am interested in what enables us to have this cognitive flexibility such that it's not tied to specific stimuli and specific contexts. And, you go to a different grocery store, like how can you be much more efficient across all, right? And we use this behavioral paradigm to model after this type of cognitive flexibility. And here the question is there such a fast representation to be retrieved and help behavior? And the answer is yes. However, it's not as abstract as we think or we hope it could be. There is rapid retrieval of conjunctive representation, but it's not as the extreme abstractness that we hope to have, which also makes sense because, maybe depending on the exact context, you don't want it to be the same and then make the mistake. Because on the other hand of this automaticity, we also have the great flexibility to switch around, right? So this kind of conjunctive representation we think is a stimulus, control state, rule, and then response representation, but it is still quite bottom-up driven and quite specific to the stimulus, the context that you acquire it initially. And it actually fits well with the abstractness Fusi and other colleagues have been looking at in the monkey physiology data in prefrontal cortex. And surprisingly, they also did not see a complete abstractness encoding of all these different contexts and things in prefrontal cortex, but they saw it in hippocampus. But anyone who's interested in learning more about this abstractness can check out their papers. Okay, so that's all I have. And the work was done with a undergrad. He was a CS undergrad major, so he was good at implementing all these SVN and but through this project, he actually got interested in cognitive neuroscience. So I was very proud of being able to mentor him. All right, so that's all I have.
Great. Thank you. Thank you very much. Very interesting, and I think it will be a good thing to look at your future results in these directions in a year or two. Definitely worth of it. So we have two questions.
Okay.
The first one is from Maxwell Murphy. I'm wondering about the switch relationship to the decision-making whereas binding of the motor intent related to the correct response. On the switch task, do you use the same two physical buttons to respond, or are they different physical buttons?
Yeah, so the two tasks share two buttons, and that is to create some conflict too, so that when they do have to switch task, there is a little bit like the task to response mapping. So in our stimulus set, there are some stimulus that's always tied to the same buttons across two tasks, those congruent ones. And so for a while, I thought that maybe I should exclude all those. But then, reviewer thought it was too strange to have imbalance of that. But in any case, to keep things simple, that's what we have. We didn't have two different response sets and everything. We want to kind of maximize the overlap between tasks so that it's slightly more difficult, but at the same time keep it simple so that we don't have to worry about modality and all that. I don't know if that answered the question. What was I? Oh, the other thing was other research have suggested that even though through many trials, people could learn some kind of shortcuts so that they know this stimulus in this task is this button, and in that task is the other button. But given the trial amount we have to the subject, we don't think that's the case. We think they still go through the task to figure out what's the correct response. So the other criticism often we receive is, how is this different from a straight SR kind of learning, if that makes sense?
So that makes sense. Thank you. That's a response from Maxwell. And another question we have. So on page 12 of your presentation, what ERP waves do you think contribute to difference between high and low switch probability ERPs? It's 12, page 12.
Yeah, this one.
I think it's the same you have on this page here. It looks no, maybe not, maybe different.
So right now, or I don't know if the question is about the representation or about like this tiny difference. Can you see my slides, right? No.
Currently, no.
Let me see, because I think the sharing is stuck with this page. So does the question is about this 12. One? This one.
I guess next one probably, not
This
like
one.
that.
Okay.
Yeah,
Like
this one.
what difference?
Correct.
What allow it to be accurate decoding? Yeah.
Correct.
So if we look at actually the projected SVM weights tell you a little bit more. I think because this shows that it's actually all over the place. So for each subject, they could build some kind of representation that's a little bit stimulus specific. You know, the butterfly with a particular switching probability. And the answer is I don't know exactly what's the what's the exact representation, but that these two are different, if that helps a little bit. And if the question is more about, we see a difference in switch cost behavior, and it can be driven by either faster switching or slower repeats. And that's also a muddy place. I don't have a straight answer. In some paradigm, we do see at a group level a faster switching at the high items. Other times we see slower repeat on the low items because we always kind of look at them as a contrast. We don't always have a baseline to say, this really is the high switch probability item faster switching versus the low switch probability items slower repeat. Yeah, sorry, it's all nitty-gritty details of the laboratory task. It's very different from the first two presentation, which yeah.
That's interesting. Thank you. So we have no other questions. And Eugene, thank you so much for presenting and for being so kind to join our conference today.
Thank you for inviting. I'm happy to share.
Thank you. I'm glad to invite next two speakers from Carnegie-Mellon University. I want to thank them for being so initiative and Dr. Douglas Weber for being so responsive and eagerly joining our conference. And it's my pleasure to introduce Dr. Serhii Bahdasariants. So far I know he's deep in computational modeling of motor and neural functions. So far I understand his main application of this modeling is application for rehabilitation of neural pathologies. So Serhii, it's my pleasure to invite you, and we'll be glad to hear your presentation.
Thank you. Thanks so much for inviting. It's a pleasure to share my research with this community. I also find the presentations today very interesting. Let me try to share my screen really quickly. All right. I assume you can see the full screen, right?
Yes.
All right. Thank you. All right. So we'll shift the gears just a little bit. We'll try to continue the same theme of how can we describe different pathologies with different techniques, and hopefully we'll get you excited with the way we do it. I'll talk a little bit about the neuromechanical approaches or modeling approaches to study some of the diseases, and we will probably start big picture. So one thing that's got me really excited to study human motor control and sensation is just realization of how complex the human body is. Most of us saw a variation of this picture, either ourselves in a museum or somewhere on the internet. It shows you the progression of the complexity of how human body developed. So one thing that not many of us think about casually is that human body has about 244 degrees of freedom. And what I mean by degrees of freedom, if you look at your hand, for example, if you start moving your phalange independently, that's 1 degree of freedom. Second degree of freedom, you can move your wrist, elbow, shoulder independently. So in total, there are 244. Just to give you a perspective of why that is so exciting, when you hear folks talking on the internet, on the news about controlling the rocket and how hard it is to land it on a platform, we're talking about control of 6 degrees of freedom. The rocket can move along the XYZ axis and rotate around those axes. So what we're going to talk about today is not quite rocket science. It's much more complex than that. And every one of you controls something like that. But the thing I want you to appreciate from this slide is we have so much more degrees of freedom that are strictly necessary for us to enact simple daily tasks. And what I mean by that is if you imagine a very simple thing that you might have done this morning. When we reach towards the cup, every reach of ours is pretty much the same. The end of our hand is tracing the straight path. The velocity is bell-shaped. We accelerate and then decelerate towards object not to bump into it. But the thing is, you could have been using so many more different strategies of how to reach to that cup. You could oddly tilt your body to the sides and you could apply many, many more other strategies. Now, in addition to that, again, as I said, somehow we optimized it evolutionarily to perform almost the same movements. From classical electrophysiological experiments, we know that when you reach from one point to another, the end point of your hand reaches in a straight path. As I also said, the velocity of the movement is bell-shaped. You first accelerate, almost throwing your hand at the target, and then decelerate to interact with it in a more precise manner. And also the joint angles, which just the way we describe the motion in an easy way, are also sigmoidal over here. So now what I want you to understand from this slide is somehow the central nervous system selects one movement strategy from many, many, many more allowed to us. And there are also so many muscles that can be used to achieve exactly the same trajectory. All right, so how do we think central nervous system does it? Because it does it somehow. One of the first theories by Feldman was that we are reflective and reactive creatures. The idea is that on a level of muscles, when you stretch them, they start resisting. That resistance is controlled by the level of stretch reflexes that sit in your back and the spinal cord. And those stretch reflexes modulated by your brain on other supraspinal regions. The idea was that that central command that I'm showing you here is just some threshold or equilibrium point command that tells your muscles, hey, to be in this position over here, the activity of the muscles should be at this threshold. Now, the problem with that theory, as we realized later, is that sensory feedback that you get from moving your arm somewhere or from bumping into something is too slow. You need to deliver the motor command and then receive the outcome and process it. Now we know when, for example, somebody throws a ball at us or any other object, what we do is we first look at it for a little bit and try to analyze how it will end or land in our hand. So if somebody throws a ball from a ceiling towards you, what you start doing is you start preforming your hands to catch it and also tense up the muscles a little bit. To prove it, here is some data from classical studies which show that before the impact of the ball, which I show with the red line over here, your muscle activity starts ramping up in an effort to stiffen your joint and not allow your hand to yield too much under the weight of the ball. So what I'm trying to say in here is we use some predictive strategies when we move as well. So the conclusion is central nervous system combines the feedback that we get from interacting with objects with some feed-forward control, which is very important for how we move, how we escape from predators evolutionarily, and other things we do during the daily life. Now, what about that feed-forward control? How does it work? Usually we think of this control in terms of two models, and you already start associating with the title of the presentation. I'll be mostly talking about models and their use. So we think that central nervous system somehow constructs a model of ourselves, meaning that it knows how the hand will move given this force or this twitch of a muscle. So it understands your body. In the block scheme like idea is we think of a desired movement. So say reach towards the cup. We take into account the physics of our body, the properties of our muscles, and we produce a motor command that goes down from the supraspinal regions to the muscles to activate them. Now, we also think of a forward model. Forward model is something we need to start adjusting our understanding of the body. So it takes the same motor command, passes the same representation of the body, and then creates a predicted behavior. Now, the error between predicted behavior and the desired behavior just used to adjust your model. So one of the contributions to the field that is about, from our group that is about to be published is we described all of these transformations that go into the forward modeling and also at the inverse if you inverse this pipeline. As you can see, taking the neural commands, correlating the neural commands to the output movement requires you to understand the underlying transformations. We need to take into account muscular tendon transformation, which is just taking into account the properties of your muscles and how do they work together. Now, there is a physics part that we all love from high school and the first university classes where you need to describe Newtonian motion of the body and understand what forces are necessary to move your limbs. Then there are integration and other kinematic transformations, which I can talk about if we have time later. But what I want you to understand from here is all of these conversions from neural commands to the predicted behavior can be described algebraically. Now, if we can describe them, what is the problem? Why do we still not understand them with these models that I'm trying to present? So here I'm showing you a video of a simulation of a rat motion with the model on the left. All these wires over here represent muscles, and all the bones obviously represent the physical properties of each bone, some inertia, some forces necessary to actuate them. We can model each muscle, knowing its properties from back in the day. We can model it with a system of springs and dampers. Obviously, when you stretch a muscle, it resists like a spring. And if you try to do it fast, it resists like a damper. So now this introduces nonlinear dynamics that are already pretty stiff and it's hard to model them. Now, in addition to that, there is a bulk of physical equations that describe your body. Here, I'm showing you differential equations, which just means they describe the difference of something in time. So differential equations for 2 degrees of freedom. So say one phalange and a second one. As you can see over here, and as you can palpate from all of your body, we have so much more going back to those 244. Unfortunately, for that amount of degrees of freedom, analytical solutions do not exist. And it's not that we didn't find them, they quite literally do not exist. So we need to use numerical simulations. Those are very slow, and to start diagnosing diseases or starting putting out some interventions that can work in real time, we need to create models that are both fast and accurate. So this is something we were working on for quite a bit, and recently had a couple of points of success. First thing I will present you is the part with physics, where we try to create models that would be used for the gait research. And I might drop a couple of lines about the research on the arm and hand motion and function. The first thing we did is obviously we needed data to understand the gait and its progression. So we took the system called Vicon. It's just many reflective markers that put on the landmarks of the body to basically track the motion in space. We did that for the several healthy adults, just showing you results from one over here. And we created these models that I was referring to. So inverse dynamics models, so dealing with mostly the physics part of the computation that central nervous system potentially executes, and the forward dynamics model. The idea was we recorded the angles, we pass it to the models to understand what forces or torques, which are rotational forces, needed to actuate the joints, and then we apply inverse of that to bring the angles back to then understand how accurate our model is. So one of the preliminary videos that I found from a couple of years ago when this development just started is here I'm showing you an inverse model in slightly lighter color and the forward model slightly darker color. As you can see, we created an accurate model that reproduces on its own. Obviously, there is a statistics analysis to that in the paper that is published, but I'm focusing on the visual part right here. Now, okay, so we created this model. What about some errors? What is it fast enough? So the errors of predicting torques and the errors of predicting angles were less than 5% of the range of motion and maximal observed torque, which aligns with the errors that humans make. You want it to be ideal, but there are always trade-offs. And I also claimed to you that the model is real-time, or rather supposed to be real-time. Here, not to get into many of the details, this 35x and other x is just the number of times we're faster than real time. And what I mean by that is if you have a second of long recording, if you want to compute it faster than real time, 2 times faster than real time will be half a second. So as the movement is unrolling, we're able to predict the forces that are causing it or what's going to happen next. So this was what was important for us. And then next we applied it to a clinical population. So to prove that claim that these models are not only useful, but are the way to go in analyzing highly complex dynamical systems. All right, so where did we apply it to? I will focus second half of this presentation mostly on stroke, and we can talk about other things as well. So as you know, stroke is a cerebrovascular accident that happens in the brain. It can be a blood leakage that kills off the neurons, or it can be that artery got clogged up and the neurons are not getting enough nutrients to survive. What it results to in an acute phase is usually severe weakness, hemiparesis. Unfortunately, one of the family members of mine just went through the same condition, older one. But they first do not walk, do not move. And what it results to over time in a subacute or chronic phase is oftentimes this odd type of gait called stiff knee. Why is it called that? You can see that compared to quote-unquote healthy leg, this leg on the affected sides looks very stiff. It's kind of looks like a stick. And the person, in order to walk, needs to circumvent it to the side. Now, unfortunately, nobody knows, and I quite literally mean that in a not overselling way, nobody knows what is the mechanism of this stiff knee gait. Some assume this might be weakness of some muscles. Others assume it might be a stiffness of the different joints, or rather muscle tone around them. And one way to describe it is to use these models that I was just presenting to you. Now, quickly, why does nobody know what is happening there? Well, just for appreciation, very quickly, we know that in the spinal cord, the dynamics that control the motion, the neuronal dynamics, are highly nonlinear. Here, for example, you see some recording from the or rather reconstruction of neural activity in the spinal cord of a mammal, a cat, and you see this neural oscillatory activity happening in there. Now, so what I'm trying to say here is highly nonlinear, and it's also differential equations. Now, if you also add the nonlinear dynamics of our body on its own, if you look at this video here, you see a completely passive mechanical systems. There are no muscles to it, no motors to it. But if you put it at an angle where it would get energy from the gravity, and if you put it on a treadmill like that and just let it be, it finds some equilibrium and starts walking almost like we walk. Again, trying to draw your attention here that our body itself is very dynamically coupled nonlinear system. Now, so again, answering that question about the stroke, why does nobody know what's wrong with that gate? The complication comes from this nonlinear interaction between the neural dynamics from over here and the mechanical dynamics from over here. So we just can't disentangle it with the regular linear models. So that's why we need something that takes into account those computations CNS does. All right, so let's jump right into it. So what are our potential hypotheses with that stroke research? Well, the first one, the easiest one, I guess, right? If you cannot bend or flex your knee, probably there's some problem with knee flexors. The muscles just don't pull it up. Another one is maybe they're stiff or spastic knee extensors. It's just these muscles pull onto your leg, so you cannot bend it. It's always straight because of that. Well, maybe there are also weak ankle extensors. Whenever we make a step forward, we need to push off with our foot, right? And the third one, most counterintuitive one, but I want you to remember this one because it will come into place a little bit later, is weak hip flexors. So maybe, when the muscles around your hip are not pulling your leg enough, some of the whip and the interactions in the joints are not contributing enough to the knee motion. I will explain that a little bit in a little bit if that's still not intuitive enough, but you will see from the data. All right, what did we do? Who we recruited? We recruited with help of our collaborators, 20 people with stroke, which is quite a group. And we had healthy controls, obviously, to that. I have some descriptors of demographics and the level of stroke as well. So yeah, feel free to inquire about that. What did we do? You saw, kind of saw this picture when I was explaining you how we constructed the model. But here we did slightly more than just recording the motion. So we recorded it, then we computed joint angles. Joint angles is just an easier representations of motion. Instead of just having tracked some points, we say instead, oh, your knee was bending to this angle, your hip was bending at this angle. It's just an easier descriptor of motion and easier to analyze and understand. Then we also used the model that I show you that calculated torques, those rotational forces, to understand what forces led to that motion. There were active, interaction, and gravitational component. Very simply, active is the one generated by muscles. Interaction is something that comes from the another joint. Easy to understand. If I were to ask you to move your shoulder to the side very fast, as you start moving your shoulder, your elbow starts buckling automatically. This is because the force generated at the shoulder propagates to your elbow and whips it out. So this is that interaction component between the joints. And gravitational torque, very simply, is the one to oppose the gravity. If you were in space, there would be no such torque that you need to generate. So we use simulation to decompose all these forces onto components and understand, is it the muscles or is it the interaction or is it something else that led to this pathological gait? So some data to support what I was saying. We recorded those angles of the knee, computed them, and you see that in control, the excursion of the knee is much larger than in stroke, and obviously there is some stats to support that. And in the hip and ankle, the progression is also very different. All right, so what do we get from here? We see that a lot of things are different. Knees different, ankle propagates differently. There is a lot of variability because ankle dangles. Hip hip needs to be at the different angle for the knee to progress. So a lot of different things, but impossible to pinpoint what is going on mechanism-wise. So let's go try to do it. We calculated first the net forces around the knee, right? To understand what forces propelled it. So in the control cohort, you see that, yeah, there is some profiles of forces. And then in stroke, it's more variable since they differ a little bit, the way their impairment changes their gait. But you see the change in this initial amount of force that they apply. So you could say from here that the force around the knee has dropped that would probably mean for most folks that, well, there's not enough force in the muscles spanning the knee to pull the knee and generate the torque. Well, if we were to end our analysis there and do not decompose it on components of force to understand if it's muscles, if it's interaction torques or anything, we would just say, yeah, probably treat the muscles around the knee and see what happens next. So not to do this guesswork, which is pretty much what was done before with this modeling, not that it's not informative at this stage, but lacks this dimension of analysis, we decomposed on these additional components. Let's look at them. So if you look at this active component here, which is the muscle-generated torque, if you look at the beginning here, what you see is at the beginning, the knee muscle, the muscles spanning the knee, so the hamstrings and others, are actually contributing almost exact amount of force as in healthy. And obviously we have stats for that in a paper. Now, what you see where this difference is actually coming from is from this interaction from other joints. Remember, the interaction is, I gave you an example, when I ask you moving your shoulder to the side really fast, what happens is your elbow opens up or buckles just because of that interaction between the joints. So here the conclusion is, it might be not coming from that active muscle effort. It may be the interaction. So what is the story there? Let's look at the other joints. Where does that interaction coming from? All right, let's look at the hip. Same analysis. We calculated the forces around the hip, and again, there is some big difference like wasn't here. Again, we have stats for that in paper. So let's decompose again. What you see is the interaction torque here. It wasn't significantly different, but the active one, given a lot of variability as well, was very, very different. Very, I mean, significantly. So now the story is that the reduction in the forces in the muscles spanning the hip caused were the major contributor to reduction in the total torque, which then caused these problems with the knee. So the problems are not really coming from the musculature spanning the knee, it's coming from the musculature spanning the hip. Very counterintuitive. So obviously we needed to look at an ankle because it's also in this interaction chain. And most of that initial force is different in the ankle, but the story there is ankle is so small of a segment compared to the lower leg and the upper leg that it contributes not much if you look at the relative forces around the joints. Now, the story that I'm trying to tell you here is without this neuromuscular or musculoskeletal modeling, we wouldn't be able to understand what influences from the hip, from the ankle are coming and impacting that knee. And unfortunately, what is currently a standard in the field and what we're trying to push away from the local clinics is the treatment of the muscles spanning the knee itself. And as we just showed that in most cases that at least we analyzed in this particular group, the knee was not a problem, it was the hip. To recap very briefly this quick study and the importance of the models, in the normal gaits, there is this active torque around the hip and there is this interaction torque. The interaction torque about the knee is generated as a result of this active influence. So in the stroke, there is not enough of the active torque here, and thus there is not enough interaction. You can see this also on yourself. The beauty of this research is that it's applicable. You can test it immediately on yourself. As you walk later today, try to think of how much attention do you pay to the fact that you're flexing your knee. You're not really using those muscles. What you do is you flex your hip, propel your leg forward, and knee automatically buckles under that interaction torque. So to wrap up this modeling story, we're doing a lot of stuff related to the arm modeling itself that is much higher degree of freedom system. We published on that as well, patented some of the approaches that we're trying to push to Department of Defense, as well as working with some folks in Ukraine to try to advance that for cure and treatment of different diseases. And yeah, there's some more work on the physics part of this coming out soon. One bioarchive preprint from the last week, and another one is coming either today or tomorrow. Yeah, none of this would be possible without friends and colleagues from former alma mater of mine, West Virginia University. Yeah, great effort, a lot of support from different institutions, and really great to have grateful to have met these people down the road. And yeah, with that, please, any questions?
Great talk. Thank you. It's nice detective story on the scientific ground. So I wish you to finally that your suspicions on the guilty will be true.
Thanks.
So we currently don't have any specific question. Maybe they will come like in a minute. So I just want to ask a very simple question. I'm not very familiar with biomechanics, but regarding your model, do you try or does it make sense to like isolate some joints or strain some joints, something like that, to test your model?
Yeah, of course. That is a wonderful question. We are actually trying to do some of that testing. It is still works. It is still in works. We're trying to get the grant from NASA, and the idea there is, as you mentioned, some of the joints might be either constrained a little bit in a suit. And another one is there is no gravitational contribution to the joint action. So one of the components of that decomposed chain of torques will be gone. So we're trying to see the global question that we're trying to answer is how motor control changes in non-gravity environment. The importance of that comes from the observation that whenever the astronauts, cosmonauts come down from the space, they cannot quite walk. It could be due to muscle atrophy, but it also could be because some of the brainstem control rewires itself, since we don't need to counteract that gravitational torque anymore. So we do some of the studies that you mentioned. We put them in a grant, and fingers crossed we'll get it. And I can answer this question better next year.
Okay, so I will have to invite you for our next conference then.
Thanks.
Thank you. Great talk. And there is a question from Maxwell Murphy. So Maxwell, I've unmuted you, so I think you may pose this question directly.
Sure.
Feel free to do that.
Nice talk, Serhii. Thanks for that. I was just wondering, in general, you're kind of talking about this role of the hips, and clearly from the side of the interaction torques and the active torques that made a lot of sense. Are you guys also considering on the sensory side any of the reflex arcs and the range of motion for the ankle, especially those differences, making any significant contribution there as well, or is that in future work?
Of course, yeah. So just for everybody, Max is actually in the room near to mine over here, so we can keep chatting about it later today as well. But yes, the story with even the mechanoreceptor feedback is growing more complex there, because ankle push-off is not as important biomechanically in as the swing phase has been initiated already. Right, so it's not the push-off itself as a moment; it's already when it started, as we marked it as marked the start as the toe already started going up the air. But yeah, the sensation of the push-off itself and their inability to locate their foot in space because of that is a major contributor there. Now, one thing that I also wanted to add. These results are very true for the cohort that we analyzed. There are some folks that genuinely have problems with ankle push-off, and that's why they do use those active ankle foot orthosis with a little motor that gives them that additional push-off. So yeah, as with any complex disease, it needs to be analyzed on a patient-specific level if you're actually targeting the treatment. Yeah, thanks, Max.
Yep. All right, and I guess I'll jump in with sharing mine.
Yes.
I don't know if there's any other questions, Serhii.
I see no other questions. So, Serhii, thank you again. By the way, when you was talking about these bouncing effects, about journey, I was for some reason imagining some boxing game. Probably it. Okay. So I think that's it with questions. And Maxwell, thank you again. Thank you for supporting our conference. And it's my pleasure to invite Pawel Herman to present his talk. So we collaborate with Pawel for already 12 years. I'm always glad when he joins our conferences and gives a talk. Actually, I think that we are overexploiting him, trying to invite him for any conference we have. Anyway, it's always a pleasure for me to hear his presentations. I think that they have the only very huge drawback. They are limited in time. You should have like 2 hours for Pawel to present in full glory. And Pawel works on working memory models, so-called mesoscaled working memory models with his colleagues from Pawel Institute. I'm waiting for time when he will present their new theory, so-called hard call working memory theory. So his colleague Earl K. Miller from Pawel Institute was giving a talk at the previous SFN meeting. I was expecting that he will present maybe something related to that, but the talk was different. So Pawel, we will glad to hear your presentation, and the floor is yours. Thank you for joining us. So
it's always a pleasure. Thank you very much. Thank you very much for. I'm absolutely honored and very glad to be here as always. So it's really a pleasure. So don't talk about overexploitation. I feel that your extended hospitality is really paying a lot of, offering me a lot of opportunity to talk to you. So what I'm going to present today, it's some of it some of you must have seen. But I want to build, I want to build sort of a bigger theory here. And I will not show you much of the most recent theory, as Ilya mentioned, unfortunately, because I was hoping that I get my PhD student actually to give me all the plots beforehand, but it will take another 2 weeks, I see. But I'll see, and I'll try to point towards the direction. And next time, I promise to show you some of our newest plots. However, I have some new component on the episodic memory, and I'll show you how we connect some of the theories on working memory to episodic memory. But before that, I'll give you some overall overview of our approach to memory studies, so that kind of you better understand what is our ambition, what are our tools, and what type of questions we can actually ask. So let me share the screen. Here we go. Good to start from the back, isn't it? Okay. So I'm based at Sweden, and as Ilya mentioned, we do collaborate a lot with L. Miller's lab in Pawel Institute. Actually, currently there is my former PhD student who has also contributed to that work, Nicholas Christantidis, and my very good colleague who is currently in Sweden. He actually was a postdoc at L's lab, and this is actually how we established a very good functional link, and we exchanged we've written co-author quite a few papers and exchanged a lot of, I think, ideas. But yes, as Ilya mentioned, like my main scientific interest is in working memory, and most of you probably know what it is, but I always want to emphasize that working memory is not only about storage, short-term memory storage, but it's a lot about actually maintaining information and using information. So dynamically operating on information, and that dynamic aspect and flexibility is, I think, the hallmark really of working memory, not so much the short storage of memory per se. And typically would associate short-term memory or working memory with prefrontal cortex, though it's becoming more and more debatable if it's only prefrontal cortex, and even more so, actually, the extent and the anatomical exact anatomical location of prefrontal cortex itself is being even questioned. So obviously there's incoming evidence demonstrating a wider involvement of other parts of the brain. Though in our models we mostly focus on prefrontal cortex, and when we think about other parts of cortices, we immediately think about other parts of memory system, including long-term memory, episodic memory, as well as increasingly subcortical regions like hippocampus. But if you look at the memory, it's actually one of the most studied systems when it comes to cognitive neuroscience. It is considered to be heart of cognition, really. And it's been studied really from a very wide range of different perspectives. In psychology, you have lots of studies in cognitive neuroscience, neuroimaging. There's animal studies in human, computational studies. The problem is that often the evidence demonstrated by all these different studies are not always playing on the same field, or not always united under the same framework, theoretical framework that would allow us to better understand the mechanism in the brain behind the actual phenomena we observe in, for example, psychological studies. So when it comes to computational and theoretical work, it would be sin not to mention, as really the father of these first thoughts of working memory, Baddeley and Hitch. In the 70s, they had this phenomenological model, like a box model of how working memory should work. And up until today, some bits and pieces of that model functions in psychology. We do operate, however, at a slightly different level, which I'm going to say a few words about in the coming slides. So it's important for our work to remember that memory is a kind of intrinsic information processing part of the brain. It's not a part of the brain, an anatomical part of the brain. It's not a function itself only, but it's really part of this information processing capabilities of the brain in a bigger picture. And another thing important to bring up here is related to Baddeley and Hitch model, that in psychology and in cognitive science, there's been a lot of this sort of box models, descriptive models, and our attempts with computational networks wants to go and try to sort of break that trend a little bit and go and study networks themselves, because there's a certain I would say weakness in describing cognitive processes only in terms of words, rather than trying to put them in mathematical formula, and most importantly in the body of the actual networks that could be embodied in our brains. So typically, what we expect from modeling approach is that it provides us some kind of understanding. And if you use mathematical formula, if you use neural networks as a substrate of your models, and if you study neural processes at the level of neuron synapses, immediately you have a temptation to talk about mechanistic aspects. So you want to understand how these neural mechanisms come together that manifest themselves on a mesoscopic level, giving rise to phenomena observed typically through our behavior on a macro scale, or sort of macro dynamics through the neuroimaging lens. We also want our models to offer us certain hypotheses about causal relationships, because if we know our mechanisms, we can better understand what happens if we change something in the model. We have certain very specific expectations about the result of that manipulation. Therefore, this prediction power and model validation are very important aspects of modeling effort. And of course, there are various different models. Some people are very interested in studying the dynamics of the brain, which is fascinating. You can collect a lot of signals. You can study them using various different mathematical frameworks, non-dynamical theoretical frameworks. You can study them using probabilistic frameworks. But what we try to do in our work is to combine dynamics with actual functions. So we want to simulate the actual function. We want to understand how dynamics speaks, or how function speaks to the dynamics. So we want to connect those two different spheres. And the way we operate, and as you can see, as you know yourself best, brain is organized at various different levels, and one of the biggest challenges we have communicating also across different groups of scientists working on the brain. If we operate different scales, spatial, temporal scales, we sometimes find difficulties communicating across those scales, because we look at the phenomena from different perspectives. But where we operate in this hierarchy of different scales and organization levels is pretty much from the level of synapses up to the level of subsystems. And increasingly, we are interested now more and more in systems, so where we connect subsystems into actually parts of a brain as such, and then try to understand function more globally. One important aspect to add as well here is that these networks we build have a spatial extent, and that has a specific importance for some of the more recent theories, where we would like to argue that space actually plays an important role in how the dynamics leading to function is manifested in the brain. So it's not only the logical position of neurons somewhere in the abstract space, but actually it means that they're occupying very specific space, and the relationship of being close or far for neurons is actually important in the network. That's something unlike really what is typically studied in those type of memory model or network models, and as such actually also in neural network, artificial neural network fields as such, in the bigger picture. So our modeling philosophy is to stick to the biology as much as we can. Of course, we do have respect for a great level of biological detail, that if we don't know that level of detail, then we try to abstract it away. So for example, we, as I said before, we do not go deeper than a neuron and a synapse. We're not trying to really go into biomolecular machinery behind ion exchange and behind the synaptic processes, but really understand them at that level using more simplified mathematical formula. And from neurons, of course, we build networks. From networks, we build subsystems, and we connect the subsystems or subnetworks into bigger networks, like a networks of networks, trying to really follow some of the topographical, but also some of the statistical data on the connections between neurons and networks themselves. We want this process overall to be hypothesis-driven. So we don't have this ambition to try to find the largest possible pool of biological detail based on which we can connect things together and then hope for some emergence, but we have some idea of how function arises in the brain, and we try to implement this idea. So that's why it's hypothesis-driven, and then we see how well the biological detail we can fill in to make these models biologically plausible. So these models should have strong functional relevance through the hypothesis that we have, and dynamical features through the emergent phenomena arising out of the biological implementation of this machinery. And in the end, of course, there is a certain level of neural details we want to preserve so that we can connect reliably to the data. And typically here we connect to either electrophysiological data, so we talk about LFPs, spiking, but also macroscopic recordings like EMG, MEG or EEG. And then we, of course, operate on the sum of population of neurons. So at this kind of level, we want to operate and make sure that the biological details allows us to, I wouldn't like to say reliably, but at least plausibly think about simulating or synthesizing these brain signals. And overall, of course, simulations are at large scale. So for that, we typically use supercomputers. And more recently, more and more, we kind of try to rely on some neuromorphic hardware that helps us to minimize and minimize the energy cost of these simulations. So how do we approach this top-down perception, top-down idea of a function? We build on the so-called HEPS theory of cell assemblies. So he was a Canadian neuropsychologist. So that the date is actually wrong here. It should be in 40s. And his idea was that interaction between individual neurons builds into a bigger so-called cell assemblies that collectively model or at least account for mental thoughts, mental memories, or overall we can say maybe more generically mental objects. And he had also idea of a sequence of such phase sequence of such cell assemblies. He would call them various ways, but referring pretty much like a kind of train of thoughts or to thoughts as being a sequence of those kind of states. But let's focus on cell assemblies as such. They've been studying neuroscience at different levels. There've been a number of different scientists, not only in the theoretical field, but also in the experimental field. They try to identify neurons that would, for example, collaborate or fire simultaneously, addressing or accounting for these similar mental objects. And these studies have been performed both in short-term memory, long-term memory. There have been various different ways of obtaining some footprint of these neural cell assemblies using electrophysiology, but also using staining more recently in long-term memory context. It's like in long-term memory, typically is to stain, and then after post-mortem, you can actually identify which neurons were associated statistically with certain typical experimental paradigms or memories in experimental paradigms in animals. So neural assemblies, cell assemblies, or ensembles, if you talk about neural memory. So this kind of typical names people would refer to those memory objects in the brain. And this is something which we also try to build in our models. We try to basically build this collection of functional neural units that would collectively account for different mental objects, and in our case, of course, memories. The most simple or the simplest mathematical instantiation of such a model is something which neural network people know as Hopfield network. In essence, what it is just a collection of neurons which are tightly recurrently connected with each other. So here you can see on the left-hand side these three sets of neurons, A, B, and C, and the idea with this kind of complex graph is that neurons B connect to each other, neurons A connect to each other, and neurons C connect to each other. And the idea would be that neurons A, B, and C, they would, in their own groups, account for a specific mental object. And interestingly enough, if you train the systems in the right way, then you can actually enforce those in like-to-like connectivity, and then you can stimulate these neurons, and you can cause kind of the overall effect of activation in these networks, and that would be kind of what we would call a memory activation. So here's an example on the right-hand side where you have neurons A, and if you activate those neurons referring to nodes A, then you have memory A active. If you do the same with B, then you have memory B active, memory C active. So how do we then go actually from this very, I would say, simple and mathematical models to biology? So first what we need to find is we need to find the good candidate for this individual, as we call them, units, A, B, C. We need to find them good biological candidates. And against some of the different opinions, let's say, or theoretical ideas, we don't think these are individual neurons necessarily. We actually see that cortical columns in cortex, they would be much better candidate for these units. So in essence, we do not connect directly, recurrently, a large population of neurons, but we connect columns. Of course, connection between columns translates to connection to neurons, but on a pure functional level, it's actually, or more abstract level, you can see it as a connection between neurons in those columns that would account for memory system. So the column typically we would typically look into the superficial part of the columns simply because these neurons anatomically demonstrate a very wide range of connectivity across the cortical patch, horizontal connectivity. Well, that's very characteristic for superficial layers. That's why we would hypothesize that this part of the column is the one responsible for really making these heavy connections, forming cell assemblies across larger patches of cortex. So in essence, it boils down that we have this collection of columns, specifically superficial portions of neurons in those columns that connect together recurrently, forming these memories. Apart from that, we also start looking a lot into the input part of the column, so what actually feeds information to this column, which is layer 4. And more recently, we've been very interested in actually looking either deeper parts of this column, so more layer 5 and 6, and trying to better understand how they participate in the memory system. There are different hypotheses about this, and we are just playing with different ideas. We don't have any conclusive evidence yet to show, but I hope next time I can show a little bit more on this. So far, then let me just limit myself to layer 4 as the entry to these columns, and layer 2, 3 as a kind of distributor of connections across cortical columns, forming the cell assemblies in the large distributed scale. And importantly, one of the important anatomical features is that we also observe, of course, in the cortex, this local inhibition. So inhibition has some local extent, spatial extent, and that spatial extent would define what we would call a macro column or a big column in the cortex, and that would be basically the extent where the local mini column compete for activity. So this local, typically fast spiking, so-called basket cells we observe in neuroanatomy, they would have a very characteristic effect of disinhibiting some parts of the local neurons and activate, or allowing other neurons to activate. So in this kind of a ping type of network, so pyramidal inhibitor interactions, we see a lot of competition between local columns. So if one column becomes active, that immediately means that other columns surrounding this inhibitory cells become quiet. So this kind of allows the cortex to be locally selective and globally form patches of distributed cell assemblies. So we would then pack those columns, and we would consider these packed columns really as parts of the cortex, and then we simulate this using supercomputers. And here I show you like the big patch, 1.5 millimeter per 1.5 millimeter. We typically actually model smaller patches for the sake of energy constraints, but also time, because this 1.5 millimeter patch takes actually longer time. And we observe, of course, how this effect scale with spatial extent. And the conclusions we get typically in our investigations is that, up to 0.5 millimeter actually, then the scaling, at least to 1.5, 2 millimeters, doesn't really provide any extra emergent effect. And then beyond, maybe there is something interesting happening. And of course, if you have a smaller network, there's a lot of other problems, I would say, which make your network to be less biologically plausible, because to make it stable, you need to make certain sacrifices, and we don't want to do them. And as I said, if you study working memory, then you associate this patch of cortex with prefrontal cortex, but you can also well think about this as part of the long-term memory, but depending, of course, on how you tune parameters in your cortical circuits, if you make those circuits more short-term responsive or long-term responsive. So there are certain parameters that would control the dynamics of learning itself. And these networks can exhibit a lot of interesting phenomena. You can cue them, you can provide a partial or very noisy cues, and they would still end up with this kind of global distributed activity. So they're quite powerful in this respect. They also operate in oscillatory domain. So before you kind of cue them, all the neurons in the network are behaving kind of similarly. They occasionally fire, low firing rates, and the moment you introduce the cue, the firing becomes much more stimulus-driven and focused, limited to a specific subset of mini-columns, recalling this idea of distributed pattern being activated at the cost of other neurons that have been inhibited. And this will be typically attributed to oscillatory activity like gamma. Let me just skip this. I want to show you this ground and active state, and when we can skip this as well, and when we activate those specific parts of the cortex, then we get this kind of a inhibitory wave, long-term inhibitory wave, which would attribute to theta, and the gamma oscillations resulting from the local interaction between pyramidal excitatory and inhibitory cells, like a ping network, classically known. This interaction would cause gamma oscillations. And there's a lot of biological evidence demonstrating indeed that the gamma oscillations in the cortex are local and typically arise out of this local gamma beta oscillations. If we let a network without any stimulation, if we allow network to remain this more, let's say, global state without any particular memory being active, then we would have what we would call ground state, and this is associated with slower oscillations like alpha, beta oscillations. And then, of course, we make a lot of arguments and try to make comparisons to the data, and we can observe then when looking at the data, that there is a lot of actually interesting phenomena and a lot of similarities between the data itself and our model. So typically what we see is this push and pull effect between gamma and beta cycles. So we observe a lot in Miller's data is that where you have a lot of gamma oscillations, then you have very, very little beta alpha oscillations and vice versa. And this kind of goes up and down throughout the whole cycle of typical working memory experiment. So when you process intensively your memory item in memory, then you have a lot of gamma activity. When you're waiting for the stimulus to come to activate memory, or you're waiting for a cue to direct your actions during the working memory experiment, then you have a lot of more alpha and beta. You're in a kind of a rest state, waiting for things to happen. And that we observe a lot in our model. Interestingly enough, if you actually look closer, also in our model and in the data, turns out that these oscillations are really on a very short time scales. So these are not wrong persistent activity episodes, but really short-term activations, bursts, we would call them. So this burst beta and gamma seem to really, on average, make an impression of persistent activity. And this actually led to theories in 80s and 90s about so-called persistent activity footprint of working memory. This is one of the still existing theories, so there are still people who find neurons who are more persistently firing over the long periods of time when the memory is maintained. In our experiments, where, for example, there's no spatial navigation involved, we actually do not observe that. We actually see most of the neurons really being fired or being coupled to the gamma cycles on short time scales. So we see actually that it's more of an expression of this new idea of more active correlates of working memory than just this is static persistent activity throughout the maintenance period. So we see it like a more dynamic manifestation of mechanisms that allow also more flexible access to memory. So it doesn't mean that you need to actually activate your neurons for long periods of time to be able to access them, because the memory still is encoded in synaptic connections. So this gave rise to what Stokes calls silent working memory, meaning that not all working memory has to be expressed through the neural activity, but it's residing in synapse. It can be awoken by stimulus and manifest neural activity, but it doesn't have to. And there's been more and more evidence demonstrating this kind of dynamic coding, and this is very much where our thoughts have gone. So what we impose in our models is the various different biologically plausible phenomena, like synaptic augmentation, that allows neurons to fire more on a more, let's say, limited amount of time with neural adaptation, and that causes sort of the or intrinsic mode of neurons firing and then quieting down, allowing other neurons from other cell assemblies to take role over and then fire. So in some sense, we would argue that our manifestation working memory more come from the fact of like interplay between different memory items. So it's not that just all memory items are becoming active at the same time, but they occupy their time slots during the maintenance period when they become active and then become silent. And then when they become active, they refresh their synaptic traces. So they kind of help neural circuits maintain memory trace of them. We also introduced something we call fast haven plasticity, and I will not be going into detail. It's a different version of SDP. We would argue that it's a more elegant, theoretically version of the SDP. One big difference between this learning rule we introduced and the SDP is the fact that we actually, our strength of our synaptic connectivity between neuron A and B is not only defined by the strength of their coactivity, like Hyde would say that if neurons are synchronously active, their connection should strengthen. We claim that as well, but we say they should strengthen even more so if their firing is particularly unique to that coactivity. So meaning that the neurons are not active just because they're always active, right? Because that will be less unique to that specific pairing. That's why we normalize our weights, our synaptic connections by the average activity of a neuron. So if the neurons are only active in those periods of time when another neuron becomes active and they become synchronously active together on a unique basis, that's the best possible evidence demonstrating that these neurons are actually really part of the cell assembly. And that has, of course, implication for many things. So in our models, we also can generate this kind of gamma theta cycles we typically observe as correlates working memory. We see a lot of phase spike coupling effects or phase or field coupling effects across long distance, similar to the data. So a lot of kind of interesting electrophysiological phenomena collected in like early 2010. They have been quite nicely reproduced in these type of models. We've been also, of course, discussing a lot with Liessmann about his idea of theta gamma coupling. And I can also just explain that our model is a bit different than what Liessmann proposes. For those who know, Liessmann's idea would be that each theta cycle is the home for multiple working memory items. For us, however, each theta cycle is a home for multiple gamma cycles, but all these multiple 2 or 3 max gamma cycles, they would rather signal the same memory. So the next theta cycle would be another memory. That, of course, has implications for timing and for different type of predictions. And sort of kind of we're in ongoing discussion. Some of the phenomena we can better explain based on this behavioral timing than this one, some of them worse. So we sort of keep discussing these issues. One of the interesting phenomena we can reproduce also is something which is in cognitive neuroscience known as a working memory load correlates. And one of the most known correlates of how loaded our working memory is, just to also give indication of how occupied we are in our task, is the amount of gamma activity and theta activity, as well as beta. And our model predicts that the more memory items you have in the brain or in your prefrontal cortex, the stronger manifestations of theta and gamma will be, and the weaker of beta and alpha. And that's very much in line with the data. And our model actually predicts it's not necessarily kind of an amplitude modulation or time modulation, the cycles are longer of gamma or something. Our model predicts it's actually more of these beta cycles we observe, gamma cycles we observe during working memory experiments. And that's because if you have more working memory items, then obviously each of them will contribute around gamma cycles. So altogether, you pack more of these gamma cycles during the maintenance period. And that's actually in line with our this kind of predictions we confirmed later on in the data from monkeys, thanks to Miller's lab, and we could demonstrate this, prove, let's say, our hypothesis based using the data. We can also demonstrate some so-called primary recency effect from psychology. So this is where you tend to remember better the items you recalled first in a kind of a long sequence of items you were supposed to remember, or those that you actually you remembered last, so those that were the last in that sequence. And those that were kind of in the middle of that sequence that we were supposed to remember, we tend to actually not necessarily forget them, but remember them less. And statistically, this kind of gives us a U-curve demonstrating that those in the first and the last would be most or best remembered items. We can demonstrate in our model as well, and we can also argue why this is the case based on the data or based on kind of the network model. And we can clearly see that actually due to the interaction between the synaptic plasticity effects and synaptic augmentation. So it's the interplay between different time constants that allows to obtain this kind of U-curve. We have absolutely no other, unfortunately, we don't have other manipulations of the model to prove that hypothesis right. So let me just spend 2-3 minutes now in the very end to tell you about how this connects to other studies, long-term memory studies. So this fast synaptic plasticity I described before, the synaptic rule we described, it's built, of course, on some mathematical phenomenological description of biological data. But the idea of this pre- and post-synaptic firing, as you know, originally comes from the long-term memory. So if we change the time constant in our mathematical model, it turns out that this model starts functioning very well as a long-term memory storage that is capable of both remembering but also forgetting. So if we couple then short-term memory and long-term memory operating different time scales, then all of a sudden we can study very interesting effects of memory recalls happening on both short and long time scales. And when they happen in short time scales, because that's typically what we can simulate or we can do in experiments, we don't wait hours during the experiments, but at least we can understand better how our memory recalls are not only built on our working memory capabilities, but also on associations with our long-term memory storage, right? Because we cannot decouple our working memory from our long-term storage. And this kind of phenomena that became a subject of our interest over the last couple of years, and we've been very much looking into this connection between prefrontal cortex and parieto-temporal cortices. So we simulated different kind of multimodal long-term memories, and we try to understand how, for example, prefrontal cortex can be used to couple different long-term memories temporarily, and then during the extensive experience of being exposed to those memories and being reactivated by means of both prefrontal cortex and hippocampus, you can actually form a long-term binding, direct binding between those different long-term memories. And we studied this in various different paradigms. We looked also at short-term effects, how these networks couple, and we tried to, of course, reproduce again some field-to-field coupling effects in electrophysiology. Though electrophysiology in this respect was kind of, there's much less studies because it requires you record from different areas. So we also started looking here into EEG, MEG a lot, and try to understand how this long-term, for example, gamma synchronization, long-term theta coupling, how they can be explained by this type of experiments and simulations using network models. And we can again reproduce a lot of interesting phenomena there when it comes to these oscillatory effects. We found some pieces of data would also corroborate our hypothesis about how distant, like spike field coupling may arise. But again, this data is very, very rare. Ryan Camalti used to collect this data on a more regular basis. And we also make these models part of a bigger cognitive architecture, as I mentioned before. So we try to couple those memory models to more like cognitive phenomena like decision making, connected to the subcortical regions, to basal ganglia. But this is kind of a beginning of the road for us. So I don't have any specific experiments to show you, rather than idea or concept that we're currently working on. And obviously here is the extension of our working memory idea, because we would like to study now not only how we recall memories, but also how it is that we have this flexible control over what memory to pick at the right time during the task-driven behavior. And not only during those kind of very well-trained behaviors, but also in cases where we learn multiple tasks, and during the task, the strategy can change. So we would like to understand how we can quickly relearn this, because this will give us also insights into the format of these representations in memory. Because we would speculate that the representations of memory themselves, specifically in long-term memory, and how they are indexed in short-term memory, they would tell us a lot about this interaction between relevant tasks. And very recently, not so very recently, but more recently, we've been also very interested in understanding how like the working memory recall is affected by our episodic memory storage. So we try to study the interaction between the short-term scale recall and the long-term scale recall. For that, we used some data from rats episodic memory studies, very interesting studies related to olfaction, where interesting behavioral phenomena were reported, a little bit not controversial, but maybe counterintuitive, because they spoke to the power of episodic memory over working memory. And we try to understand really what is the regime, why episodic memory sometimes takes over. So we try to kind of model this in the system or distributed system with working and short-term memory, and also with episodic memory, which would be multimodal, related to olfactory cues, but also related to different contextual cues, because they also seem to have been an important factor in those experimental studies, determining whether rats remembered specific odor better in a specific time sequence or worse. So without going into detail, because we don't have time for that, I can tell you that we managed to reproduce some of this experimental data quite nicely. And it turned out that a lot of mechanisms behind this were hidden actually in the synaptic plasticity. So how these memories are formed across this different long-term modularities, like the olfactory representations and the contextual representations, as long as the short-term memory effects operating with short time scales. So all this came together and gave us kind of a nice picture that allowed us to reproduce behavioral results. There were no, unfortunately, dynamic or electrophysiological or other recordings made during that study, so we could not really, let's say, connect on that level to those pieces of data. Now we are currently looking for other sources of data to make stronger predictions about this. One interesting fact to mention here is that when we replaced our learning rule with pure SDP, this behavioral effect completely disappeared. And we kind of understand that it's actually to the fact that synaptic plasticity, according to those more traditional SDP formalism or theories, is not normalized, as I mentioned before, by the activity of neurons. So it's kind of a blind, it's not aware of the uniqueness of pair of neurons firing to a specific coactivation of related patterns. In our case, olfactory cue together with a specific contextual cue. In this experiment, contextual cue was associated with arena in which rat was found. So our model, because of the specificity of pairing, wiring neurons that are unique to that specific pair of stimuli, gave us this behavioral results. So I think it was a strong prediction for us that this model actually is meaningful. So I'm sorry for running a little bit out of time. Just as a summary, I can say that overall, I hope that I managed to convince you a bit, at least, that models are useful, that there are different ways of modeling, but this theory of attractors, the recurrent networks, is something that was worth attention, I think, in not only working, but memory studies, because it basically builds on this kind of intuitive feeling that the brain is in the flux. It's responsive to inputs, it's responsive to internal states, but ultimately also has capability to engage in the quasi-stable states every so often and move between these quasi-stable states. And these quasi-stable states would basically refer to our memories, to our perceptual understanding, to thought, to mental thought, basically. So in this framework, I think the strength of this model is that we can connect this top-down perspective of attractor of recurrent memories to the actual implementation. And that allows us to connect, in some cases at least, to the heart, electrophysiological or macroscopic recordings from the brain, and connect them to the more behavioral manifestations. So it's basically the attempt here to cross this barrier across different scales and levels of brain organizations and somehow integrate all these different biological phenomena into one theoretical framework. So with that, let me finish, and again, apologize for running a little bit out of time, and I'm absolutely happy if there are any questions.
Thank you, Pawel. Great talk. So far, it's the last presentation. So who is interested may just simply raise hand and ask directly the questions. Okay, so I would like to ask one question, as always. So, Pawel, there is, I know that it's not completely supported by all the labs and scientists, but there is opinion that there exists so-called professional or specialist memory. So if you specialize in some specific area, you usually have quick access to a wider number of concepts which you can easily manipulate with, and it stays in between the long-term memory, which is long to access but which has a very large capacity, and between working memory, which is quickly to access but which has very limited capacity. So do you think that such phenomenon like specialist memory exists? And if it exists, can it be explained in terms of your model? Thank you.
That's an excellent question, Ilya. So the term I've heard in different contexts, yes. And the way let me put it like this. I am absolutely convinced there is a capability of the brain to be more or to learn specific items or specific concepts better than others, depending on your experience. And I think that's been shown behaviorally, that even how we recognize faces, but maybe that's not a good example because there are specific circuits to do that. But our episodic memories, our experiences, they shape, of course, and our professions as well sometimes also help us to have more detailed memories. And these models, yes, they can do that. And it's mostly because of the power of associations. Because we need to remember that according to those theories, at least, memories are not static. So they're constantly reshaped, and they are shaped through the interaction with other memories. So they're not isolated kind of items. And of course, as you train, as you add more memories, and typically when we talk about the long-term memory, you change the way, of course, you memorize a specific types of memories at the cost potentially of others. And when it comes to this interaction between long-term memory and short-term memory, it's a similar story. So if you have a very strong, let's say, representative power for a specific, let's say, specialist memories, then obviously you also learn, let's say, a more detailed or a more, let's say, reliable way to recall them into short-term memory. So in other words, you'll need less specific cues to recall them, statistically speaking. And that's also something we can show in our models. If we overload, let's say, our long-term memory with kind of patterns which have very specific statistical properties, then we can see that certain cues in short-term memory are more likely to recall them. And there are different explanations. We could, of course, argue about this, that we sometimes, our thoughts kind of run away towards certain memories. And the last thing to mention, you mentioned this kind of item, this issue of being able to quickly recall. That's a slightly different thing. So how quickly you can recall things, it's partly the time it takes for you to make association active or move the association to your prefrontal cortex. So basically connect kind of your long-term memory to your kind of index in working memory, which is driven by goal-directed behavior or a specific task. So that's one thing. And the other thing is also tuning of your network. So there are neuromodulators, and they can further tune your network to either be, to operate on the scale between the speed and the accuracy. And this is a dynamical phenomenon which works beautifully with attractors, because you can demonstrate how by changing the level of excitation in these networks, you can make them sharp, like really going quickly, acting very quickly, but not necessarily targeting a very specific memory, but for example, activating a couple of memories and then they compete. Or on the other part of the scale is where you take a little more time with a cue, so you're not so fast, but you target your specific memory, so you're very accurate in terms of memory you want to activate. So of course, it's a speculation, but I see this modulatory effect of various neuromodulators providing a flavor or level of excitation to our networks that could potentially explain this kind of speed versus accuracy phenomenon or balance.
Oh, great, thank you. I think that actually in this case you should account for a very huge amount of factors which may influence that.
I can imagine. I can absolutely imagine.
Yeah, thank you.
Thank you very much.
Okay, so we don't have any other question, and thank everyone for coming. Thank you so we have our conference dedicated to the 100th anniversary of the founder of our department. So thank you for coming, for paying respect for our teacher. And I think that he will be definitely happy to know that sometime his department will host conferences like this one. Thank you.
Thank you very much again for invitation, Ilya. I want to just say as well, and lovely to see you all as well. It's fantastic. Thank you.
Great. Okay, so we'll be finishing then today. See you all.